briefly describe the process you use to decode the messages. Proteins are made by joining amino acids together into long chains, called polypeptides. The first step in decoding a message is to transcribe a nucleotide-based sequence from DNA to RNA. Then, the mRNA enters the cytoplasm and the sequence begins with the start anticodon UAC that corresponds to the methionine amino acid. After that, the anticodon is linked with its corresponding codon. The process continues until it reaches the codon stop. What role plays codons in protein synthesis? What is used coded messages? RNA contains 4 different bases: adenine, cytosine, guanine, and uracil. The genetic code is read 3 letters at a time, so that each word is 3 bases long and corresponds to a single amino acid. Each 3-letter word in mRNA is known as a codon. The synthesis of all proteins begins with the amino acid methionine. It is used to translate the nucleotide sequence of the mRNA to a protein sequence in the translation process. What 6 letters will not appear in the coded messages? There are only 20 different amino acids. This means there are 6 letters of the alphabet that don't encode a specific amino acid. The letters that are not abbreviations for any of the amino acids are B, J, O, U, X, and Z.
For a protein to be built, its blueprints first need to leave the library of DNA. Specific DNA sequences encode the protein and transcribe it into a complementary strand of mRNA. DNA and mRNA share characteristics that allow for the creation of coded messages. The characteristics involve the relationships among the different bases of DNA and how the proteins they produce can signify letters.
So for example, we have to encode this message that is in DNA to mRNA. So we have to take this chart as a reference, and T goes to A to U, C to G, and G to C. The first codon is T, so the T goes to A, the G goes to C, and the A goes to U. So we have ACU. In here we look for A, C, and then U, that it is 13. So this 13 is T, so the first letter indeed is going to be at AT. So we just continue this pattern until we have the complete message. So the message is take care. So the next message that we have to decode is this one, and it goes to DNA to mRNA. So as we did in the previous exercise, we do the same, like with this chart or table. I don't know, G to C, T to A, and A to U. So we do the same with the other columns, and it turns out that it is his spy.
So for the first message, we're going to use the codons to amino acid table, and there we are going to search for each letter in order to write a message. So the table with the changes will be like this, and the phrase will be meet me at 5. So with that done, the phrase that will be formed will be the plans has changed. In the final message with the changes already done, the message will be the ship sails now.
How do you know which databases must use when you transcribe the DNA sequence to RNA codons? Nucleotide bases are adenine, thymine, guanine, cytosine, and uracil. Adenine is always paired with thymine, or uracil, and guanine is always paired with cytosine. In transcription, a single strand of DNA is transcribed to RNA with the respective base pairing, except RNA doesn't contain thymine, instead it uses uracil. Predict the DNA sequence for the first message in part B. Begin with TGT, CGA, CGA, TGA instead. Is the message changed? Why or why not? The message will change because each base has a specific pair that it bonds with. Suppose some codons, two different amino acids, are mapped. What will be the effect on your translations of code message? What will be the effect on protein production? When some codons are mapped to two different amino acids, then the translation of code message will become inaccurate. Due to this kind of mapping, several copies of different proteins will be created, and most of them will fail to work. The protein form will have different compositions, since there is not a single amino acid given to the codons, but this kind of mapping never takes place.
During the real production of proteins in a cell, what could happen to an RNA chain before leaving the nucleus? Before it leaves the nucleus, the RNA is spliced or cut to create mature RNA. This splicing involves that cutting out of introns, leaving only the exons, which are the segments which code for a protein. What step could you add after transcribing the DNA to develop a more complete model of protein synthesis? After the transcription of the DNA is complete, the genes encoding the amino acids for the proteins are copied onto the mRNA standard. After this, the RNA standard moves out of the nucleus. The ribosome attaches the mRNA standard to itself, and the tRNA reads to the codon and brings the amino acid to the ribosome and attaches it. The process is continued till the protein is formed. This process is known as translation.